the way the mice are playing. They're messing around till they all start swaying. Down in the club where the joint is jumping, the birds and the bees and the hip chicks are swinging. There's a cool cat in town, never settled down. She loves chasing the dogs around. There's a new kid around, and she knows she is bound to be the leader of the whole rat pack, and she won't wait for the return of the Mac. There's a cool cat in town, never going down. One day she's even heading for the crown. A cool cat in town. Show the big dogs what to do. Zip-a-doo-da-ba-dee-a-ba-doo-bop, ba-doo-dee-a-bo-ba-dee-bop, zap-a-ba-doo-bop-ba-dee-oh-bop-bop. Zip-a-dee-da-zap-zap-a-doo-bop, bop-bop-dee-da-bop, zip-a-dee-bop-ba-dee-da-ba-doo-bop. Play cats. There's a cool cat in town. Oh, believe me, no cat's cool as this kitty cat. She won't wait for a new dog around. Who don't like some cats in town? A cool kid around. A cool cat in town. Oh yeah, baby.
Yes, hello. I'm Gail Ouellette, President and Scientific Director of the Regroupement Québécois des Maladies Orphelines, the Quebec Coalition of Orphan Disorders. We're sorry for starting late today. We have a guest from the United States, and there's some technical problems today, but we'll start. And Just to an introduction to, this is our before last Zebinar, as we call them, during our month of February for for the month of rare diseases, and our theme this year is the Diagnostic Odyssey. So all our presentations have something to do with the Diagnostic Odyssey, but we're very happy today to have Ms. Sharon Terry, who's the President and CEO of Genetic Alliance in the United States, and she will also be talking about many of their initiatives at Genetic Alliance. So to introduce Sharon, where she's also, besides the President and CEO of Genetic Alliance, she's a co-founder of the Genetic Alliance Registry and Biobank, and also of the Platform for Engaging Everyone Responsibly, the peer program. She's an example of a dedicated parent of children who have a rare disease, and dedicated. She got really involved in trying to advance knowledge, research, and towards development of therapy for the disorder her children, who are now adults, are affected by. The disease being pseudoxanthoma elasticum, and she actually co-founded what we call the PXC International, which is a research advocacy organization. She has a master's degree in theology, but by creating that PXC International and working for the disorder of her children, well, she worked, and even in the research aspect, she co-discovered the ABCC6 gene, which was patented to ensure ethical stewardship, and assigned the rights to the foundation. She subsequently developed a diagnostic test and conducts clinical trials. She is author of 150 peer-reviewed papers, of which 30 are clinical PXC studies. She's involved in many major international and national organizations. I don't know if you heard about the big initiatives in the United States for precision medicine and cancer moonshot, but she's involved in those initiatives. Also, accelerating medicines. She's also on the advisory council of the National Center for Accelerating Translational Science at NIH, on the Roundtable on Genetics and Precision Health at Global Alliance for Genomics and Health, and in the International Rare Disease Research Consortium. She is also on editorial boards of several journals, and one of them which is an official journal of Genetic Alliance, the Genetic Testing and Molecular Biomarkers Journal. And she led the coalition in the United States that was instrumental in the passage of the Genetic Information Non-Discrimination Act. And she received an honorary doctorate from Iona College in 2006. She has had many other distinguished awards, and I'll let you read her bio on Genetic Alliance's site. She has been honored many times for her work in genetics and rare disease space. Just before I give her the mic, I just want to tell you that you can always write any questions in the Q&A at the bottom of your screen, and for the francophones also, there's a translation, and you can put your questions in the Q&R at the bottom of your screen. So I'll let you speak, Sharon. Thank you very much for accepting to present today, in spite of the technical glitches you've experienced. Your mic is still closed, as I see.
Great. Yes, you so patient being started. Okay, can you hear me now?
Yes, it seems to break. Go on again, and we'll see if it's. audible.
Can you hear me now?
Yeah. Yep.
Can you hear me now? Oh,
Yes, that seems great.
okay. Okay. All right. So this may be quite challenging, and I apologize for being late. I apologize for the technology difficulties. Some are not ours. There's servers that are apparently down for Teams in the U.S. And I'm on a cell phone trying to do this, so we will have to bear with it. I'm very happy to present in Quebec because my relatives come from there, my father, and so happy to be with you all. You can go to the next slide. I'm going to experience a delay. I think I don't see the next slide. Let's see if I can maybe run it on my computer.
There it is.
Okay, great. There we go. Okay, great. Thank you. So I am coming to you from Genetic Alliance, which is a coalition of advocacy organizations started by Victor McCusick, who's the kind of granddaddy of human genetics, and Joan Weiss, wonderful social worker. We have a lot of partners and funders and have been over these 36 years growing like crazy because the world is finally recognizing that people and families and communities are really where we should be focused. Next slide, please. Gail, can you tell me which slide? Okay, so as Gail mentioned, my children were diagnosed in 1994 with a rare genetic disease, pseudoxanthoma elasticum, and we set off to do research on this condition. Next slide. And what we did is essentially as parents, and as Gail said, I have a master's in theology, but we borrowed lab space, collected blood and tissue, and discovered the gene, and you can see lots of other things that we have been involved in since, believing that people really could take charge of research. Next slide. And because a lot of families came to me asking, can you do the same thing? Can you create a registry for our disease? Can you create a biobank? Can you get an IRB together? I joined Genetic Alliance, which was already 10 years old when I joined it about 25 years ago. And what I realized is that what we could do for one disease, we could do for all diseases. And so with the permission of my children, because I knew I would slow down the research on their disease if I turned to all diseases, I did join Genetic Alliance and started to work very hard to get this done for all disease. And you can click through the next slide. Simply, I have a TED Talk to tell the story of that transition if you want to Google Sharon Terry and TED, and then you can go to the next slide after that one. So then the next, as Gail said, I've written a lot of papers because I believe that I need to be able to talk to scientists as well as to families. The families don't care about these papers, but the scientists do. And so in trying to speak the language of scientists, I have learned, I think, enough to be able to put together papers and to really see how do we not just have people-driven research be a checkbox or an idea, but an actual reality. Next slide. And so I'm going to describe to you some of the tools, and you can click straight through this slide is fine. I think we have a little lag on the slides, at least on my end.
Tu peux cliquer encore, je pense.
I'm going to,
Okay.
and the next slide, and then the next slide. So I'm going to talk to you about a few of the tools of Genetic Alliance. And so on the next slide here, you'll see a lot of different tools, and I'll describe a couple of these as we go. So you can go to the next slide. And first, I'll talk about something called Advocacy Atlas. So many times the undiagnosed particularly, but even the diagnosed have a long journey, both in getting a diagnosis, which is what the focus of my talk today will be, but also in just practical things. And we realize that while the medical system takes care of perhaps some of the difficulties that the families have in terms of the health care, there are lots of other things like financial assistance and educational services and so on. And so this little Candyland map is clickable on our website and will bring you to all kinds of resources. And they're pretty global. There certainly are a large number of US ones, but we are a global organization, and so we list any services we can find when we find them. If you go to the next slide, We've written something called Wiki Advocacy, and it's 2,000 pages of a wiki talking about how do we advance research on rare and common disease. We're really agnostic to whether something's rare or common. While I know the focus of this is rare, we are genetic disease. We also believe that probably all common diseases are rare once we stratify them with genes. And so we're really writing to families, communities, clinicians here about how to break down barriers, how to set up research systems, and so forth. So this is a rich resource, and it's contributed to by the community, so it's really a good place for people to come. And then if you click to the next slide, we have something called Disease Info Search, and Disease Info Search lists 10,000 diseases and their subtypes. It is a crowdsourced resource. It's the only crowdsourced resource in the world. All of the other directories of diseases are curated by an expert. We believe that the advocacy groups, their leaders, are really the experts, and so they're the ones who are entering all the information and data into this system. The system also dovetails with clinicaltrials.gov and PubMed, and it filters all that so that the newly diagnosed can come here and learn about their disease, but not with so much of the noise. For example, if you looked in PubMed, you'd find thousands of articles on a particular condition, and we bring those through with a filter that helps people to see the most current and the most useful ones to them. And if you go on to the next slide, you can see that we do have integrations with lots of other systems. We also have an API so that other organizations can use these data. So for example, Global Genes Directory is actually this disease info search, albeit they call it by another name. And if you go to the next slide, you can see some of the information about our statistics around how much activity we have, again, used a lot by individuals who are newly diagnosed, but also clinicians, genetic counselors, geneticists, pediatricians, and so forth. Okay, we're going to go on to the next slide and talk about PEER, which is our promise for engaging everyone responsibly, and then to the next slide, which will say that Luna promotes people from subjects of research to partners in discovery to transform research. They're a partner that we decided was perfect for us for our registry system. Over many years, Genetic Alliance developed these, and I'll talk about that a little bit more. If you go to the next slide, with this really beautiful system that they've created, and Luna DNA is owned by the people, and you can click another click, it's owned by the people who share the data. So it is the only people owned, and I'm not sure if you can click again, I'm not sure why this slide isn't showing, at least for me. Great, thank you. It's the only system in the world where the people own the company. So the company is actually owned by the people who share the data. They're actual shareholders in the company, and they get shares in the company by how much data they share, not by whether or not their data is more interesting or useful, because we really want to be egalitarian here. And essentially what we allow is DNA files like 23andMe and panels from Invitae or GeneDx and so forth, or electronic health records in the U.S. We connect to 700,000 portals in the U.S., and we are working on a Canadian integration right now, which will be almost as challenging as the U.S. You don't quite have 700,000 different brands of Epic and Cerner, but you do still have a complex system. And then also surveys. And so you can go on and click to the next slide, where we describe that we have lots of off-the-shelf surveys. All of the validated instruments we could possibly find on the planet are in here. So everything that NIH has curated, as well as many other systems around the globe. And then you can make custom surveys quite easily with a survey builder that actually helps you to constrain the fields and so forth as you need to. And in addition, all of the data is standardized and quality controlled as it comes through. So the system is built so that each individual community, whether it's a big umbrella community, so some of them are whole countries are using this, in some cases states and so forth, and then in other cases just umbrellas of diseases like all XYZ diseases are using this because the data basis is actually not siloed. In other words, as individuals enter these data, they are put into a common database with a common data model, and all of that information is cross-conditioned, cross-pathologies, cross-phenotypes, and so on, allowing a great deal of survey of analysis to be done. If you go to the next slide, each individual has a dashboard, and the dashboard shows how many shares they've earned, also shows what surveys they've taken, and so on, so that they could follow along. The administrators have their own dashboard so that they can see what's happening in their community. There's a recontact tool built into the system so that you can recontact those who might have not finished some surveys and so forth. And there's also a workbench, which I'll talk about on the next slide. So we realized, and Luna with us, that we need to bridge between the communities and the researchers. And so you can click again. And so today, communities, we believe, are the source of truth, and they really understand the issues. And so we work a lot with communities to help them understand what their members need. And then if you click twice more, communities' critical mass can validate the group experience. And Luna really accelerates this. For example, we can do the equivalent of a Delphi process in about 6 weeks, which is phenomenal and for much, much less money. And then if you click, I think, twice more, yep, and then one more time, and one more time, sorry. We understand that researchers need real-world understanding of people's health states. They want multimodal longitudinal data at a scale generated outside of a clinical system. So, for example, real-world data collection through mobile devices and so on, longitudinal information. In fact, we call this not a database, but a data stream because it's updated in some cases every day. We also realize the global consumer privacy laws are changing at a rapid pace. GDPR is only the tip of the iceberg. We have now the California Consumer Protection Act that's more stringent than GDPR. Some other states in the U.S., I know Canada is considering a number of privacy laws and has enacted some. Brazil has one, and so on. And so Luna becomes the data controller under the GDPR kind of language and takes care of all of that for us. The biggest difference with this system and others is that in true GDPR or privacy land language, we've embraced that privacy, those privacy issues, and don't see them as an obstacle to research. Because if each individual must keep control of their data, must have a string on the data as how we put it, and be able to pull it back and allow it to be used and so on, it means that the data cannot be spread around the way it once was. And so exported data passed around between researcher to researcher is not permissible under these laws. And in fact, secondary use of data is not even allowed in countries like Germany. And this system keeps the data in one place. The researchers come to the data stream, and they can do all the fishing and exploration they want to do in that data stream, and have this data updated regularly. Of course, the data can be exported for things like regulatory submissions and journal submissions and so forth. But in any case, what we've developed is a system that we believe really accelerates research at the same time as protecting people and giving them the controls they need. If you go to the next slide, I'll talk just for, keep going. The Genetic Alliance IRB. Well, the things that we were doing in 1995 and 6 were avant-garde at the time. And so we developed our own IRB because we needed a people-centered IRB. And if you click to the next slide, we have a number of individuals involved in this. Dr. Christine Carter's our chair, and then you'll recognize some of the other people here, the mix of both community members as well as scientists and ethicists, and they've reviewed an enormous number of protocols over the last however many years now, since 2003, so we are almost at 19 years. Go on to the next slide. Genetic Alliance also has a real strong interest in the neonatal, newborn, and maternal health experience, and so we run another division called Expecting Health. And in this division, we are really giving people information around newborn screening, around what it means to live with a child with special needs and so on, and trying to package information such that it's accessible and available to people where they are, where they live, and so on. And then the next slide, I will speak about I Hope Genetic Health, which is our newest and most exciting right now adventure. It really stitches together everything I just showed you, and that's why I showed you what we have already done first. And if you go on to the next slide, I Hope is established because we believe every individual deserves a diagnosis, is connected to appropriate care and research, and control their own genomic and health data. So you can see it brings together a number of the things that we have done so far. And if you go to the next slide, these are things you probably know, that there are about 250 million people with a genetic disease worldwide, 7,000 plus genetic disorders. And when you look at the data on how long does the diagnostic odyssey last, some papers say 5 years, some say 7, some say 9. We decided it's really between 5 and infinity because some people never get a diagnosis. Next slide. Another comparison that's important is a lot of times we hear, oh, but malaria is more prevalent and of concern, HIV is of more concern. But if we look at this number of 250 million with a genetic disease and 212 million malarial cases, we've exceeded that, and 40 million HIV cases. It doesn't mean that malaria and HIV are not to be paid attention to, but it does mean that even as the Gates Foundation, for example, does a fabulous job in Africa with malaria and HIV and other infectious disease, then the largest killer of children in Africa becomes genetic diseases. And so, while again, we all know they are zebras, but there are a herd of zebras, and maybe there's a different word for herd. But if we go to the next slide, I just want to share with you 2 stories about the impact of I Hope. So I Hope is a program of Illumina, the company that does most of the whole genome sequencing in the world, or at least backs the scientists who do it. And I Hope has been doing this for several years. This is one story of a 12-year-old boy in Peru, early onset generalized dystonia, hyperreflexia, knee pain, cramping, and so on. He was the only one in his family with something going on. They didn't know what. He was in a diagnostic odyssey, found a base pair deletion, also based on the clinical whole genome results. They could treat him with a already existing drug, and he was all of a sudden, quite quickly, a boy who could run and play. Next slide. We have another story here of a mother and daughter. The mother had suffered for 30 years undiagnosed, again, put into the I Hope program. The 2 of them, they both received clinical whole genome sequencing. The daughter had also been experiencing a lot of the same things as the mother, albeit younger and not as bad. They were diagnosed with Warburg-Sinati syndrome and were patients 7 and 8, treated experimentally with an off-the-shelf chemotherapeutic. The painting on the right next to the picture of the mother and daughter is a painting the daughter did about 6 weeks after being put on the drug. Her vision was restored, and she is actually doing quite well, having had a couple surgeries to correct some other things now that they've stabilized the disease. So point being with both of these cases is that the diagnostic odyssey could be ended when they did receive clinical whole genome sequencing. And the exciting thing about I Hope is that we are going to be doing that for everyone who needs it in under-resourced areas. So next slide. Okay, so this is what comes from the Illumina background that makes us aware and confident that we can do what we need to do. So they've sequenced more than 1,100 people between 2016 and 2021, most of them in the last few years. In more than 68% of the cases, they found variants. This took care of the naysayers who said, really, you're not going to find anything. This is not useful, and particularly not in low-resourced areas. They described several genetic disorders and so forth, and change management in up to half of the cases, which is very critical to us. So the Illumina program pretty much stops at the interpretation, giving back the result. We're going to actually be involved with the people, helping to connect them to either clinical information for them or research projects. And the Illumina program has 25 active clinical sites and nonprofit partners globally. So we're going to build on those and be able to expand very, very quickly. In the next slide, you'll see a report from the United States Government Accountability Office. The importance of this is not so much that this is telling you all something new. You already know all these problems on the path toward diagnosis. But the fact that the U.S. government studied this and now believes it too, and the Congress has been educated about it, is an important thing. So in the next slide, I show you our team. We've just assembled. One person starting today. The rest started last week and the week before. So we are brand new, hot off the press. In fact, this is the first public talk that includes iHope. We have a very diverse team because of the locations that we'll be working. Illumina has asked us to give about a third of our resources that we will raise to Africa, and other places we will work are Latin America, South Asia, and then North America, where people are in need and don't have access. In the next slide, you'll see that our target is about 40,000 patients per year. And those of you who know genetics know that in most cases we will be doing trios. And so we're looking at 3 times that number or somewhere around that per year. How are we going to do that, you might say? That's an ambitious goal. And it's not ambitious enough for me because it still would take us too long to diagnose the people who need diagnosis today, particularly children and babies. And so what you're seeing here is that we will be putting together clinical support, DNA sequencing, and IT and logistics. And iHope will contract with whole genome sequencing labs that are capable of doing thousands of clinical whole genome sequences, CLIA CAP approval in the US, other regulatory oversight in other countries, some the same, some different, held to the highest standards. Each of those labs will apply for a grant of reagents, and if they need them, sequencers from iHope. They will come to us in an RFP process with clinics already named, and then we will contract with both the labs and the clinics, because one of the very important pieces of this program is that the results are returned to the people, and those results are deposited in the Luna platform to allow the people to control the data, and also to allow that data to be completely openly accessible with the permission of the individuals to every researcher on the planet. And I like to say, and perhaps smart Girl Scouts who will be able to suss out things that scientists don't have time for, because with this many conditions, we don't have enough geneticists, obviously. And then once the individuals are diagnosed, we'll be working with those individuals, and we get to work with them even de-identified because of the recontact tool within Luna, which will allow us to give them connections to already existing pharmaceuticals. We are building a pharma consortium that will give access to drugs in places where that's not possible, or we'll work with the insurers to get that drug paid for. And then for the 40% or so of individuals, 30% that don't have any variant found, or that need more research toward drug development, therapy development, we will be supporting the clinical research that needs to happen, because the platform allows, again, any researcher to come into the platform. Academic scientists and nonprofit ones will be completely free, and the cost for pharmaceutical companies is relatively low compared to lots of other systems where they have to pay CROs and others to get this information. So this whole system we will be bringing together in the next couple of months. We should have an RFI, so a request for information, out in a few. Oh, could you please go to the next slide? Sorry. That would have helped for you to see how this looks. So anyway, I described it. And if you'll go to the next slide, this has all been made possible by a grant of $120 million of in-kind reagents and machines for use in under-resourced areas. And you can see Genetic Alliance, Illumina, our peer program, and Luna all in collaboration here. This is great, but we think, to raise about another $250 million in cash to be able to pay the labs to do this work, to pay the clinicians to do the work, to get the families on a bus to the clinic and so forth. We've been in conversation with a lot of partners, including the Gates Foundation that has some good on-the-ground infrastructure. They would very much like to be a partner in this program. AWS, Amazon Web Services, will provide for free all of the storage and computing that we need. And then we have conversations begun with lots of other major entities that you could imagine, like Google and Microsoft and so on. Okay. And then the next slide simply says that I am very aware that I need to raise this much money. It's become one of my major endeavors, and that I need technology and pharma partners and so forth. And so we are on a big push to go forward with this, and also looking for small donors as well. I've raised lots of money with $25 and $30 kinds of donations, and we'll need a little more here, but we will be able to do this, I believe. There's such great enthusiasm, and people finally get that this is really, the technology's ready, the people are ready, and so forth. And you can go to the next slide, and I can say thank you very much for your time, and again, sorry about the technology glitches.
Thank you very much, Sharon, for that presentation, especially that new initiative, I Hope Health Initiative. Obviously, that those are all the things we're looking for, and presenting this month of February with our theme on Diagnostic Odyssey. I was happy you had your photo there on your slides, because we missed you there in the video, and it's nice to see you now. Yeah. I have one question, and then we'll see if we have questions from the audience. Well, I want to say first that we all know that the underdeveloped countries, we say here in Canada that we don't have any official yet program strategy plan for rare diseases, but we do have a universal health system, and people do get care, and it's just that we would like to advance better on management, on diagnosis, on research. But we know the underdeveloped countries, especially these days with the the COVID and lack of access to vaccines for many countries. So your initiative and your bringing it to these underdeveloped countries is really nice, very nice initiatives. We can wait second if you help out people who need it more. So, and one question. Yes. So there's a link between PEER, your registry platform, databases, and this initiative. I was wondering, in your PEER program, do you already have data in there from the undiagnosed? And is that the link that's going to be between the genetics and the phenotyping, that is the describing of these undiagnosed cases?
Yes, so we do have some. However, this will be until now, I would say there are certainly some people who have genomic information and phenotypic information. Some people. Putting 40,000 a year in, and I believe it will be more, I think that's our kind of low bar, will greatly enhance that system. And I think dovetailing with existing systems, like I love Phenotyps, which comes out of Canada, with Kim Boycott and others. I love those kinds of programs that have already attempted to do this. We believe that those sorts of things will be juiced by this. This will be the kind of enormous amount of data that will allow both obvious kinds of conditions that are already available to be diagnosed, but also the undiagnosed conditions as well, because matching phenotypic data in the Luna system has built this phenomenal workbench with many, many tools that allow those kinds of associations to be developed, which is why I joked that, Girl Scouts or moms and dads will be able to stay up at night and do this, because it will be like a lot of other, there's a lot of citizen science compute projects in physics and in astronomy and ornithology. This is going to be like that. That, again, the people's identity is protected, the data is there, let's explore, let's see what we find. And so, yes, there is undiagnosed individuals' data in Luna, but there's a lot more diagnosed data individuals in Luna right now because communities use it as the platform for their registries. But when we start to pour this data in, it will be quite remarkable.
That's great. And well, I've been telling all our presenters the last week who were presenting on genome sequencing, exome sequencing projects that I don't know if you saw it in Nature in the last week, an editorial, a letter about data sharing across the world. And that's the only thing that's going to make us advance to getting diagnosis as rapid as possible, because we need all that genomic sharing with all the variants, and instead of working in silos. So I guess you're for such an initiative.
Absolutely, yes. And we believe that lots of times that data sharing happens with de-identified information. And for us, that really leaves out the person in a really dramatic way, in the sense that you don't know what happened next week, next month, next year. And the person also doesn't get back results. And so this is really novel what we have done, and we realize that doing the messy thing of keeping the people connected to the project the entire way and allowing us essentially to be able to give them back results and so on, and encourage them to be part of other research programs. It also democratizes the data in a phenomenal way, in that no one is going to have a leg up over anyone else. Now, that when I speak to scientists, they get very worried, saying, oh, but I need to embargo the data so someone else doesn't publish and so on. Absolutely, and we have systems built in to do that, following guidelines that have been set by world bodies around data sharing, so that, yes, of course, you can embargo the data so that you can write a paper, do the analysis, all that stuff, do drug development, which would take years, and that's also fine. But I think what's novel here is that the ability to keep this connected to the individuals. They are not, tokenized. We can tokenize it, so you can bring in an external data set and match it up with the folks that are in there, but we really want to see a robust connection with the individuals.
Okay. And can you tell us more about, I'm still waiting for questions from the audience, but can you tell us more about, downstream? So how is this going to work to target doing some, screening to develop some therapies?
Yeah, that's a fabulous question.
Pharma?
Yes, yeah. So pharma, of course, is very eager, and crassly put, when pharma's blockbuster models failed, rare diseases was the place they went, and they believe that's a good place to be. However, low ends, finding the patients and so forth is hard. This is going to make it really easy. We're creating a massive data stream that they could easily identify people in for specific projects. And so a lot of them have agreed that doing a lot of this, like at first, and then there will be more diseases than anybody has time to develop therapies for. So there won't be a race to develop one therapy or another. It will be more about making sure that there's enough quality data, individuals that then can enroll in actual clinical trials in clinical sites. A lot of this, our concern, and the staff that we hired knows how to do this in country, in Africa and Latin America and South Asia, because we don't want to be being typical Americans and just coming in, helicoptering in and plucking the samples out and doing the analysis in the US and advancing the careers of scientists. That is not our goal. And in fact, one of the things we believe we'll be able to do is both put some pressure on the payer systems, the insurance systems, and also on the price of drugs, because we will have in this consortium some agreements we can be making up ahead of any research that's being done. And so that's pretty exciting. And quite a few of the pharmaceutical companies, the large major ones, are very interested in this. I think they know the future lies in this sort of activity. And so it'll be, in a sense, doing some of the function of a CRO, so identifying patients in, 3 countries in Africa, and then helping with the clinical sites that we've already had relationships with to build those clinical trials with the pharmaceutical companies. And the other thing we like to think about is, in many of these countries, in South America, in Africa, we have leaped over analog technology. They went straight to digital phones. They didn't have analog phones. They didn't have analog systems. And I think we can do the same thing around the technology for sequencing, around the diagnostic therapies and so on, and also in advocacy. You know, I think we've had a long, in some sense, slog for 50 years in advocacy in North America. And in these countries, it's new. It's just being born, and they can jump over a lot of the problems and a lot of the mistakes that we made. And so I think on all fronts, it will be quite exciting to come into these areas. And the other thing is, there's a lot of work that's been done by H3 Africa, which is a program of the NIH, has at least 17 sites in Africa. They've all been dedicated to research, so they can do research use only whole genome sequencing, for example. And so the upgrade here will be that they'll move to clinical sequencing. There has also been some knocks against what we're trying to do, saying you can't do it in places like Africa and Latin America without sending, you know, hordes of geneticists to these countries. And essentially what the Illumina program did is they had geneticists go for a day to a place, for example, Todos Santos, to teach the pediatricians there what they needed to know to help them identify cases that were undiagnosed and could benefit from clinical whole genome sequencing. Those clinicians learned very fast what they needed, and then they did telemedicine for a lot of the results. And so we've gotten offers from Mayo, Genome Medical, you know, to do some of that work in other countries until we train up enough specialists in the countries. So that the people are not left alone and the clinicians are not left unaided. But we were shocked at the result that they've had of those 1,100 kids in underserved areas being served by the normal clinical system and the clinical system learning as it went.
That's wonderful. It's really nice to hear about initiatives in other countries than those that we maybe read about more in scientific publications. So, one last question. We do have people that have contacted us, our iRare Center here in Quebec, about Pseudoxanthoma elasticum. So because you're the expert, can you just, for the last minutes, give us an update on those clinical trials going on, or research in clinical trials?
Yeah, thank you for asking that. I don't ever get to talk about PXE, and of course, you know, with two children who are now working, one at Luna and one helping with some of the infrastructure at Deloitte. So PXE is in a great place. However, it took far too long, which it does, I know. 26 years of working on this. Yes, so we discovered only in the last few years that the gene is actually predominantly expressed in the liver, causes low pyrophosphate levels in the blood. And so now we have TNAP inhibitors, pyrophosphate, an enzyme that's been discovered, all of which are working in that ATP pyrophosphate pathway. Interplay with a couple other genes, which, you know, again brings us into this is not a monogenic disease the way all the rare diseases probably will wind up. And so we're in phase 1 clinical trials for about 3 different things, and phase 2 for 2 of them. Really exciting year. So while I have to raise $250 million for Genetic Alliance to do this program, I've got to raise millions to run these clinical trials on the PXE side. So I'm in a world that I've never been in before. And I figure if I could learn all this science, I could learn what it means to raise money. It's not in my DNA, but it's going to have to be.
Yeah, well, I guess any leader of nonprofits has to go into philanthropy and whatever all the time. Well, it's good to hear that the Gates Foundation is maybe on board with your initiative. They're doing, as you said, wonderful things for infectious diseases, and so it's good news if they can come on board for genetic disorders around the world. That's great. So thank you very much, Sharon, for being here today. I know it was difficult with your traveling and with some technology crashes in the United States, but at least we got the gist of your talk, and we were very happy to have you. Thank you very much.
Thank you for the invitation, Gail. Keep up the good work, and thanks for your patience with the technological issues.
You too, keep up the good work. Thank you. Bye-bye.
Bye-bye.